So in the previous chapter, we looked at what is gene expression and how RNA is made and protein is made. But in this chapter, we are going to look at how that gene expression is regulated. So when we say regulated, it means, are you going to stop the gene expression or make it active in one time and slow down in another time, or make it express highly in one tissue versus another tissue? So these are the ways that the cells or the organism regulate. But in this chapter, before we get into detail, we had also understand how prokaryotes such as bacteria differ in gene expression compared to eukaryotes. So the prokaryotes, as we have seen before, they don't have much time. So they got to make a decision pretty quick, turn on or turn off gene expression, or combine multiple genes under the control of a single promoter. So prokaryotic genes are controlled as operon, or that are called units of gene expression. So operon includes multiple genes under the control of one promoter. So in addition to the promoter, they also have another control region called as operator. So these both promoter and the operator are regulatory sequences that the prokaryotic transcription factors recognize, and RNA polymerase will bind and then make the RNA. So when that promoter or operator is bound by the protein, depending on what protein is bound, a promoter by the transcription factors that activate transcription, operator is bound by a repressor protein that downregulate or inhibit gene expression, so the transcription will happen. Then you need to think about the logic as to when do you need the genes to be expressed. So you will see two types of operon. One is called inducible operon, another one is irrepressible operon. Inducible operon is where you don't need this all the time, but when you need something, then you need to turn it on. Okay? The example of inducible operon is lactose operon, or called lac operon. So the bacteria normally don't come around lactose, that only when you're exposed to milk or milk products you have lactose. So they need to turn on those genes only when lactose is present. That's called as the inducible operon. On the other hand, some operons are turned on all the time. You need to make amino acids that are essential for the bacterium, such as tryptophan. So that is happening all the time, but when you have too much of tryptophan, then you need to control or stop the gene expression or making of RNA and protein. That is called repressible operon. The repressible operon example is trip operon, or making tryptophan and that is called tryptophan biosynthesis genes, and that is controlled in a negative manner called negative feedback, means too much tryptophan, stop it. On the other hand, lac operon is inducible. You have lactose, you induce the expression called positive feedback. So that is the general idea about prokaryotic gene regulation. Eukaryotic gene regulation is a little bit more complex. For the simple reason, eukaryotes like us are higher plants or animals. They are multicellular in nature. They have different types of tissues, and they all start from a single fertilized egg, which zygote into multiple tissues after they divide and differentiate, and that results in a differential gene expression based on the tissue. So they have to also account for the growth and development of the organism. So some genes are expressed when they are at the young, at childhood stage, or early stages of the growth and development, or some genes are expressed when they're older and more mature. So this is another challenge the eukaryotes face. Okay? The third type is where you have seen introns being present as intervening regions in the RNA. That needs to be removed. So after the RNA is made, they need to process that. So these are some of the challenges eukaryotes face. They also have nucleus where RNA synthesis and processing happen, cytoplasm where protein synthesis and processing happen. So they need to manage that. So eukaryotic genome also is much, much larger compared to prokaryotes. So if you take an E. coli bacterium of 5 million base pairs, an eukaryotic genome like humans, it can have 3.2 billion base pairs. Like even a single chromosome can be several hundred million base pairs long. How is this organized? How is this compacted is something important to understand. So the DNA has a double helix. It is just a bases and a base pair with each other is very stable form, but that wraps around proteins called histones to make a structure called as nucleosome. And those nucleosome again wrap around each other and coil onto each other, creating a structure called chromatin. And the chromatin, they are fairly uniform, it's referred to as a euchromatin. They're highly variable, it's called as a heterochromatin, and they condense into form a metaphase chromosome. So this type of compaction is important before cell division. During prophase, they go through this type of compaction. But when the gene need to be expressed, they need to uncompact. They need to disassemble and relax the chromosome into chromatin and make the DNA double helix accessible to the protein, such as transcription factors, and express the gene to make RNA. So this type of control at the long term is called as the transcriptional regulation, or the long term, where the chromosome can be compacted and not expressed at all. Or the chromosome could be methylated and the proteins cannot recognize, will not transcribe. So these are all long-term regulation. But once the chromosome is relaxed and the DNA is accessible to the enzyme to separate, make the RNA out of that, then there are transcription factors regulate the gene expression at the short-term level. So that is at the short-term level of transcriptional regulation. But once the RNA is made, then what? So some RNA is there stable for many days. But some RNA is not stable even for minutes, they get degraded. So the stability of mRNA is one type of regulation, post-transcriptional level, or there are multiple exons present. The exons could be shuffled in different ways. That is called as an alternate splicing. So this could be at the post-transcriptional level. So once that is done, the next level of control in eukaryote is called translational level control, where the translation could happen when the right time comes in. Okay? The egg has mRNA ready to go. Until a sperm cell will come and fertilize that egg, then it starts to make the protein. Or the cofactors such as heme is needed to make hemoglobin protein. Without that hemoglobin, mRNA will be there, ready to go, but not enough heme to combine. When the protein is made, it will not translate. That is a translational level of control. Lastly, we go into post-translational level of control where the protein is made, but it needs to be processed, such as zymogen activation like we saw in the previous chapter, or glycosylation, myristylation. This type of modification should happen, and that is at the post-translational level. So in this chapter, we looked at what is gene expression and what is the regulation of gene expression and what elements are involved. In terms of DNA, you have regulatory sequences, promoter, enhancer, or activator region, or repressor region called operator region. And there are proteins that bind to this, such as transcription factors, repressor protein, or activator protein. So these are the protein that bind to this DNA sequence, regulatory sequence, combined with the RNA polymerase to control gene expression. Eukaryotes are a little bit more complex. The regulation happens at multiple levels: transcriptional level, post-transcriptional, translational, and post-translational. So try to understand, organize this information, and remember the examples we use in this chapter. So this is the summary of regulation of gene expression.